Hi, everybody. This is Sam Stambula from the CCEM. I'm our outreach coordinator. I'd like to welcome you all to our third webinar series, this one being on scanning electron microscopy basics. And that's being presented by Chris Butcher, one of our SEM specialists at the CCEM. Just before we start, I'd like to point out, if you have any questions throughout the presentation, put them in the chat, and I'll be going through those and making a list of the questions that have been asked. And then at the end of the presentation, we'll take 5 or 10 minutes to ask Chris some questions. And if we don't have time for all the questions, we will put them in a Q&A on our YouTube channel afterwards. Also, this presentation is being recorded, so in about a week or two, it will be posted to our YouTube channel if you'd like to review it again. And if you're having any technical difficulties or any questions, also feel free to put that in the chat specifically to me, and I can help you out. So thank you for joining us, and let's get started. Are you ready, Chris?
Yep, let's get started. So my name's Chris Butcher. Today we're going to be talking about scanning electron microscopy. This can be viewed as sort of an introduction to the subject. I'm one of the technical staff at the Canadian Centre for Electron Microscopy. Have 35 years of experience with SEM and also a lot of experience in sample preparation for both SEM and TEM, as well as extensive experience with EDS and EBSD. Okay, so let's get started. So today we're going to be talking about a few subjects involving SEM. So we're going to be talking briefly about the types of SEMs, the components that are in an SEM, and the things that are used for that. We're also going to be talking about the electron beam interactions which occur when the electron beam strikes the sample and other objects within the microscope. We're going to be talking about some of the electrons that are produced, so secondary electrons, backscattered electrons. We're also going to be talking about the X-rays that are generated by the primary beam hitting objects. I'm going to go briefly into sample preparation, but in no way is it really going to be covering the subject matter. It's more of a sort of introduction to it. And then we're going to be talking about doing imaging in the SEM and some examples of some of the things you can do in the SEM. But in no way it covers sort of the wide range of things that you can actually do. See a nice picture here for one of our SEMs showing a catalyst at fairly high magnification. So I would generally categorize the sort of three categories for sort of general SEMs. There are, though, sometimes machines that are more dedicated to particular purposes. So for instance, you might have a machine that is an SEM, but it's more dedicated to doing some sort of analysis rather than more of a general machine that you can use for a lot of different types of samples. So recent introduction is tabletop SEMs, where you can look at a variety of samples and you have a lot of flexibility where you can sort of put the SEM. So anything from your office to a lab, or even people transport it around in vans and things like that. You have your conventional SEM, where you may have an area room, which is set up for having it. And then we have the FIB devices. So it has both an ion column and a SEM column. So you can do focused ion beam work, where you can actually mill away material and produce samples for analyzing, say, electronic circuits, or making TEM samples, doing 3D work. So quite a useful device. The two main ones from that are both gallium ion columns, and you also have plasma fibs that quite often use xenon. Now, if we look at the actual SEM itself, it's composed of a lot of parts. The main component of the SEM would be the electron column. And if you can see my thing, let's see if we can get a pointer showing up. So you have the electron column where you have the gun, and then you have the other components, which consist of a condenser lens system, an objective lens system. You have apertures for controlling beam size, scanning coils for scanning the electron beam, stigmation coils for correcting for astigmatism, and you have various detectors. So here you can see one of the detectors used for detecting secondary electrons. And you have a stage, which you'll have a holder, which will hold the sample. And you'll have a vacuum system, so it might consist of different vacuum systems to get the pressure down to a level where you can actually do some imaging. So SEMs generally operate from a few hundred volts to up to 30 kilovolts. They also will have different size sort of chambers for fitting in the samples. So depending on what sort of SEM you have, you may be able to fit only small samples in or large samples. Is very dependent on the particular SEM. Now with the stage, you can adjust the position of the sample, moving it in the X, Y, and Z or height direction. You can also rotate and tilt the sample. So here we can see on the left-hand side, inside one of the chambers of one of our SEMs, there's a sample in the holder and the stage here. So it's a piezoelectric stage. You can also see some of the detectors. So here you can see one of the EDS detectors. This is the bottom of the pole piece or the electron column. Over here on the 7000, you can see the various detectors that are present. So we have an EDS detector up here. At the back here, we have a EBSD detector. And at the side here, we have an insertable backscatter electron detector. Now, with this particular microscope, we also have 4 computers. So we have the SEM computer, an EDS, EBSD computer. I'm going to explain what all the acronyms stand for in a moment. And we have a computer for doing electron beam lithography and a support computer for the SEM. Now, when you have a sample in the SEM, you want to make sure that, for one thing, that you don't hit anything. So it's quite often useful to have an infrared camera that you can look inside the chamber. And also, it's very useful to have an image of your sample so that you can navigate around and actually position the beam where you want to look at a particular sample. So if you have multiple samples, it's very useful to have an image of the sample that you can navigate around. Here you can see one of our service rooms and one of the scroll pumps for pumping down the microscope, a chiller for cooling down the lenses, and a UPS for short power interruptions. So one of the important parts of an SEM is the electron gun. So there are different types of SEMs when it comes to guns. So the sort of general categories, you'll have a tungsten filament SEM. Tends to be fairly large source for the electrons. You then have the boride filament, a little smaller. And then you get into the field emission guns, which you have a cold field emission, which uses a tungsten source, and a Shockley tungsten zirconium oxide single crystal source. So this operates at room temperature. This is heated up a little bit. So these are our field emission guns. We have fairly good resolution with these things. Generally, they're kept on all the time. And these are the ones that are used for doing high resolution imaging. One of the problems with the cold field emission gun is that there has to be flashover done every once in a while, depending on the age of the system and how clean it is. So you may have to spend some time waiting for it to do that, and it may interrupt the sort of work you're doing. This very long time for where you can do acquisition, so very suitable for doing things like electron beam, electron backscattered diffraction, and anything that's going to take a long period of time to do. And here you can see some of the shapes of the different sort of filaments that go inside the gun here. So one of the important things, because we are looking at things in a vacuum for the most part, we have all sorts of different types of pumps that we might use to pump down the chamber. And one of the other things that's important is that quite often in the SEMs we'll have different vacuum zones within the SEM. So you'll have the specimen chamber. You may have an intermediate zone where you're pumping out things, and you may have a different pressure where the actual gun is itself. So here we have a display showing one of the variations of SEM. So this is from an environmental SEM. So we can actually have various different gases and moisture going into the SEM chamber. And we can use that for doing some experimentation, or we can use it for removing the charge that builds up on samples when we're doing that sort of work. So the vacuum, we may have apertures at different locations that help us maintain a certain vacuum level within the different parts of the microscope. And here you can see on the left-hand side an image of some of the rotary pumps that are sort of parked in behind one of our SEMs. And I have a picture showing an astronaut in a vacuum chamber, because one of the important things to remember when we are doing SEM is that water and low boiling point solvents will boil below room temperature in the vacuum. So we have to be very careful with what sort of samples we put in, and that they don't outgas and overwhelm the vacuum system. And maybe using some low vacuum or ESEM in cases where we do have things that have the potential to outgas. So to get the sample into the SEM, we have two ways of doing that. We can open up a door and basically put it directly onto the stage, or we can use an exchange chamber where we keep the SEM under vacuum, and we place the sample in an airlock, and we pump down the airlock, and then we insert in the sample into the chamber, and then take the rod back out and close the exchange chamber door, so we can maintain the vacuum inside the microscope. Very useful for not having to pump down a very large chamber microscope, so you can quickly exchange samples. Now, we have various different interactions that occur when the electron beam hits objects, and hopefully we're mostly hitting our specimen. So we have secondary electrons, which are produced by inelastic collisions. They tend to be fairly low energy. So they're subject to various fields very easily, so magnetic and electrostatic. We have backscattered electrons, which are produced by elastic collisions. We have Auger electrons, which are very electrons coming from the very near surface, which you can have a sort of dedicated system for doing Auger spectroscopy. And we have X-rays produced by these inelastic collisions. So we're getting inner shell vacancies and having the higher shell, higher energy electrons drop into these vacancies and emitting some energy. We have cathode luminescence. Now, not a lot of people have detectors for doing this. It tends to be more in geology and things to do with electronics. Light is produced in anywhere from the UV to near IR range in certain materials, semiconductors and minerals for the most part. There is some organic materials that also do this. You also may have an electronic circuit that when the beam actually hits it, it actually produces some current. So there are some systems available where you can actually image the areas that the current is flowing through. And it's very useful to see the image as well as an idea of where the actual current is flowing. So very useful for electronics. Now, when we're talking about electron beams, it's important to note that we're applying a potential to the electrons. So when we set the microscope to 20 keV, we're producing 20 keV electrons. So when we're talking about the actual microscope, we're talking about keV. When we're talking about the electrons, we're talking about keV. So what's important is when the electron beam hits the sample, we're getting all the things we've seen in the previous slide being produced. Now, you can do some calculations. I'm not going to show you any math here, but you can do some calculations and figure out, based on the energy of the electrons going in and the particular material, what the depth the electrons go to. So that's how far they go in. But what's important when you're doing detection is them actually escaping from the sample. So secondary electrons, which are the electrons that are produced by the inelastic collisions with the sample surface and other surfaces, are only coming from a very shallow range. 50 nanometers, I think, is the deepest they supposedly come from, but I think it's much less than that. Backscattered electrons come from much deeper than secondaries. And you also have X-rays. But in all cases, you're not getting anything coming from deeper than 10 microns in any sort of material, depending on what its atomic number is and its density. And this is all controlled by the kV of the beam that you're applying to the sample. Now, you can use various different formulas, or you can use some simulations to sort of calculate these sort of volumes that might occur within your material, especially if it's a little more complex with multiple layers, using programs such as Casino. Now, if we're talking about secondary electrons, there are various methods of detecting them. So here we have a nice diagram showing us two of the detectors that are normally used for detecting secondaries. So the beam comes down here in sort of a spiraling electron cloud. And we get interactions with the sample producing secondary electrons. So the SE1s are produced by the primary beam. The SE2s are produced by the backscattered electrons that might be bouncing around within the sample or the microscope. They tend to be less of a contribution than the SE1s. And then you have the SE3s that are coming from other surfaces within the microscope. So this will contribute to noise and other things like that. There are SE4s, but they tend to make up a very small portion, a few percentages of the noise that you're getting in the system. They tend to be from further up in the column of the microscope, from various apertures and things like that. So we have two primary detectors used for detecting secondary electrons. So we have the Eberhard-Thornley. It has a Faraday cage or a collector on the outside and a scintillator here. And in the back here we have a light tube, and back here we have a photomultiplier tube for actually detecting the electron signal. So as the beam scans across the sample, you will get from each sort of pixel or point of scanning, you will get a point of data which makes up your image. Now in the through-the-lens detector, there's actually a charge applied to the tube inside the column here, and there's a mirror which bounces the electrons that are pulled up into the detector here. Now for low vacuum and SEM, there's also a detector that will work in low vacuum situations or in environmental situations where you have basically more of a gas in here in the chamber, and we can actually detect the secondary electrons here. Now here we can see two images produced by secondary electrons that we've collected. So if you're looking at the Eberhard-Thornley detector, here we can see an image here. Here we can see an image through the lens detector. Now, one of the features that's very important with secondary electron imaging is the visualization of topography. So you can see that we have different sort of objects here or different sort of surfaces. So here we're getting sort of edge contrast. So you can see where we have sort of raised ridges or very thin things protruding. We're getting a higher level of secondary electron emission. So we're seeing sort of bright edges on things. So very important, seeing the bright edges on things to define the position of objects. Also, more visual in sort of directional illumination, because the Eberhard-Thornley detector is in a particular position within the microscope. You get sort of directional illumination. You can bias the collector or Faraday cage in front of the detector in order to vary the collection of secondaries within the microscope. So you can have it so it's fully on, so it's pulling secondaries from various locations, even around corners. Or you can have it turned off so that only the secondaries going into the detector directly are detected. Now, the other electrons that are detected within the microscope are backscatter electrons. So we have some different types of methods of detecting the backscatter electrons. Primarily, we use semiconductor detectors, which may consist of quadrants. So you may have 4 quadrants, you may have a dual quadrant detector. It's sitting below the pole piece here under the objective lens. The beam goes through it. It may be an insertable detector, or you may have a scintillator which fits underneath where you can collect the backscattered electrons. There's also concentric ring detectors which allow you to also detect that. Now you can vary how you mix the signal with these different detectors, where you have the quadrants or the segments. So it can be useful for giving you different types of contrast mechanisms within the microscope to see various different things. Now, another type of thing you can do as well within the thing is crystallography, where we have a camera here with a phosphor screen. So what we're doing is the beam is hitting the sample, and the backscattered electrons hitting the screen are producing a pattern, which we can use to analyze the crystallography of the material. So quite useful in doing things. So if we look down here, we can see some images and some data that we can collect with backscattered electrons. So here we're seeing some compositional contrast. You can also show things based on density. So here we're looking at the average Z number of the surface here and collecting the backscattered electrons that come from the sample. So the very heavy tungsten carbide particles are showing up very bright. The nickel matrix is showing up much darker. And in cases where you may have voids or surface contamination, you're having things show up much darker. So things that have no material or are low atomic number materials will show up darker. Here we're seeing a copper sample, and we're seeing orientation contrast. So here we're actually seeing the different grain structures or orientation of the individual unit cells within the material are giving us some contrast because of their different orientations. Down at the bottom here, we have a pattern produced by electron backscatter diffraction, which we can then, this is a pattern from a point on the sample. If we scan an area, we can then collect all these patterns and analyze them, figure out the crystallography of a material. So here we can see an EBSD phase map where we've identified the different crystalline phases within a material to see how things are distributed. Over here we have a technique which is not necessarily available to everyone, is where we can do electron channeling contrast imaging. And we can actually look at the dislocations and image them within a material. So when we're talking about the X-rays that are produced, there are two main methods for looking at X-rays. We have energy dispersive analysis of X-rays, so an EDS detector, so top image. And we have a WDS detector for imaging, for collecting X-rays as well. So main difference between the two things is that here we're doing a sort of individual spectrum with the energy dispersive analysis. Wavelength dispersive analysis, we tend to be doing, maybe up to a dozen elements, and it's a little bit slower, but it has better energy resolution than EDS, and we need standards. We may use standardless analysis in the EDS method. Can be quite useful for getting quick work on things. But both these techniques, we have a characteristic sort of fingerprint to each sort of element. We do have certain X-ray peaks that overlap with each other, so we sometimes have difficulty in separating things. WDS tends to be better because of its energy resolution in separating peaks than EDS. We can't tell anything about the valence states of the materials. There are other methods for doing that. We also cannot detect any X-rays from hydrogen and helium due to their atomic structure. Lithium, if you have the right equipment and conditions, you can detect lithium. Generally, for elements that are below sodium, an atomic number, you need to have a lot more present in your material to really detect it. And we're looking at levels of around, say, 0.1 weight percent as sort of our upper limit for sort of detecting things. Tends to be a little better for heavier elements, and lighter elements tends to be less than that. So here we can see a typical EDS spectrum where we can identify the various elements present. So we get a whole long list of things. This sort of thing you can collect in a matter of seconds, depending on the size of your detector. Smaller detectors, you have to take a little bit longer for collecting data. So today there are some quite large detectors that are being developed, and also sometimes multiple detectors are used to collect EDS data at the same time, so you don't have like shadowing effects, shorter collection times. So we can also see here the backscatter electron image of the sample we're looking at, and we can see a distribution of the aluminum within the sample, and we can see a distribution of the zirconium within the sample. So we can also, showing up here is, we can analyze different points in the sample and collect spectrums. You can also do things like line scans as well, and things quite useful. Now, when we're talking about samples, we can put a huge range of different types of things in the SEM. So inorganic, metals, semiconductors, insulators. In the case of insulators, we have to be careful about effects where we get the beam producing charging in the material. So what happens is we have a charge buildup in the insulator, and it affects the low energy secondaries. If it's really bad, it affects the actual primary beam itself. As I mentioned before, we're putting things in a vacuum. So if it's got some water in it, we have to make sure it's dry, or we have to switch to either low vacuum SEM or environmental SEM to look at things. And the same applies to organic material. So organic material will be non-conductive for the most part. So we have to make sure it's dried and we don't have any solvents present. But we do have a problem with conductivity here. So you may have to use low vacuum or environmental if you don't want to coat it, or do some treatment of the material. And when we're talking about organic material with water, mostly biological, we may have to do some dehydration in conventional SEM, or use low vacuum or environmental. So as I said, we have to consider what the effect will have with the vacuum and the electron beam on the sample. So you can get heating occurring. So you can have bond breaking occurring when the beam hits things. One of the things we quite often use in the microscope are conductive pastes to sort of improve the conductivity of materials. So joining the material to a substrate or the holder such that we get a path to ground. So primary thing when you're doing sample preparation is to know that the sample will actually fit in the microscope. So we have had cases where people have brought samples which are too large to actually fit in the SEM. So you may have to cut them down to size. You also have to consider what sort of work you're going to be doing in the SEM. So you may have to have them sized down to an optimum so that you can do the analysis you want to do or the imaging, depending on the microscope you're using. So microscopes will have different size chambers. So it's important ahead of time to consider this. Now, when you're actually coming about to put the sample onto either a substrate or in a holder, you want to make sure it's secured. So you could physically secure it, or you can use conductive adhesives and tapes for doing that. And as I said, and I will stress, we have to have a path to ground. So the stage of the microscope is grounded so that we don't have a charge building up on the stage. So you have to have a pathway to the stage where you're drawing away the electrons that might be building up in the sample. So you have to consider if you're putting something in, say, a plastic and polishing it and etching it, we have to make sure that the metal sample in your plastic has a pathway to ground. So we may have to use some paste to sort of connect up the sample in the plastic to the holder, and then that the holder connects to the stage, and we get a pathway where we can conduct away any electrons that might build up in the sample. The other thing you have to consider is that whatever preparation you do, you're going to have a nice representative sample. So it's not going to do things like obscure what you're going to want to look at. It's not going to add any elements that are going to be bad for doing sort of analysis of things. So we have to make sure that we're not, in the case of, say, biological material, if we're doing things like freeze drying in order to get the material water-free, that we don't produce any sort of ice crystals that sort of damage the structure that we actually want to look at. So quite often with biological material, you'll go through different processes to actually dehydrate the sample. It may be solvent substitution. It may be a method by which you're basically sublimating off the water in a slow process. You can also do things where you're actually cooling down the sample, as in sort of cryo stage. You can also do the environmental SEM, or you may even have a holder which you can have the environment that you want to look in a sealed sort of chamber that you can image through a window in the SEM. Quite often with biological material, you also want to fix it, basically kill it off and get it stuck down to a substrate. And you may want to do some staining or some labeling that's conductive so that you can actually see with some nice contrast the structures you want to look at. Conductive coating with carbon and various metals is very useful if you're doing conventional SEM. You can still do it in low vacuum and ASEM, but might not be necessarily required for things. So when we're actually looking at things, we're actually scanning or rastering the electron beam on the sample, and then we're displaying it on the monitor. So the smaller the area we sort of scan, the improved sort of resolution. So there's various things you can do to improve the resolution. So higher KV, you tend to get better resolution. Lower probe current, you get better resolution. If you're able to change the aperture, you can go to a smaller aperture. and do things like that. One of the biggest problems that people have when doing SEM that we see when we're teaching people SEM is the problem of astigmatism. So distortion of the image, which occurs due to possibly charging effects, lenses not being ideal for imaging. So quite often when we start off, we're focusing the beam on the sample, so going up and down with focus. And when we see stretching, we know that we have some astigmatism in the image, or if we see a sort of blurriness to things. So here we can see under focus, over focused, and the middle image is showing it focused, but because we still have some stigmation, it's also possible we haven't quite aligned the microscope as well. We need to do some adjustment of stigmation. There are some other things that will affect what you're looking at as well. So with very bad astigmatism, you will see circular objects or rounded objects look like lines. So when you get into a case like that, it's very important to basically set the XY stigmators back to 0. So here we can see we've done a little bit of stigmation, so we've improved a little bit from this image. Oh, not so far. We've done a little bit of stigmation in combination with focusing. And here, so we're doing maybe the X stigmator. And here we've adjusted the Y stigmator, a little bit of focusing, and we've stigmated the image so that we've corrected for the blurring or the elongation that we're seeing. Now, one of the other things that's important, it is a scanning electron microscope. So when you're taking images, it's important what sort of scanning rate you're doing. So it's important the sort of pixel dwell time you have on the material. So especially at lower probe currents, you're not going to have a lot of signal. So you have to be careful in that you don't do too fast a scanning rate when you're doing an image. You get a very sort of noisy image. Here, at a slower scanning rate, you get a much crisper sort of image. Now here we can sort of see some line scans overlapping the images. So here we're doing a very high contrast image. So you can see on the line scan that we're varying from white at the top to black at the bottom. And we are getting some areas which are totally black, so there's no sort of detail. We're also getting a lot of areas where it's sort of pure white. So we're not getting sort of any detail in there. And there you can see nice edge contrast around the sample. Here we have lower contrast, but we also have a less bright image. So you want to sort of adjust in between these sort of two levels to get a nice image when it comes to brightness and contrast. Now, one thing that quite often happens in the microscope, we may have dirt on the sample, so we can end up with getting contamination. So here we have a nice clean sample where we're not getting any sort of contamination. Here we've focused on a smaller area to do focusing and stigmation. And because there's either organic material or floating around or organic material on the surface, we've actually cracked the organic material and are depositing carbon on the surface as carbon squares. So you can see here that we use 3 different magnifications, so different levels of carbon deposition depending on the time. And you can see that the surface here is fairly dirty. You can see lots of low atomic number material is showing up as fairly dark in the secondary electron image. Now, if we're in the backscatter electron image, we might not necessarily see this low atomic material because of the depth that backscatter go to. You might not be imaging sort of things that are on the surface. Whereas the secondary, which are fairly shallow depth, you do tend to see the contamination that you have on the surface. And here we can see we have a contaminated sample when we've actually burnt off the contamination on the surface instead of depositing it with the electron beam. So it is important to have fairly clean samples within your SEM. Now over here we can see a plastic. And where we've been scanning the beam, we've actually been damaging the structure. So you can get bond breaking, amorphitization of materials. You can also basically evaporate things as well. So we can also maybe make a trench with the beam. So you have to be very careful about sort of the KVs that you use when you're working with different sort of materials. So one of the biggest problems we have besides stigmation is charging. So here you can see a polymeric material, various fibers. We're getting sort of bright charging here. We're getting some, maybe some dark charging in some of the other areas. So in the bright charging, we're getting more emission of secondary electrons. In the dark charging, we're actually not getting the secondary electrons going to the detector. So we really can't do an image under these circumstances. You quite often will see sort of lines or bands of noise within the structure when you have charging effects. Another effect that you'll see is you might get drifting of the image. So it actually looks like the sample is actually moving within the microscope. But what's actually happening is you're sort of displacing the secondary electron image as the charge builds up on the sample. You can do things like reducing the KV or reducing the current of the beam to reduce sort of charging effects. Or you can go to low vacuum or environmental SEMs to not have to do that. Or you can do things like coating, staining to improve the conductivity. Now here you can see an example of where we're using channel charging to in the bottom left-hand corner to actually image inside the microscope. So we're actually, the sample is charging so much that it's actually a mirror and is actually bouncing the beam up into the chamber, so we can actually image the bottom of the pole piece. And the secondary electron detector, you can see the collector grid there, and the capped EDS or X-ray detector here. Just show a few examples here. So here we can see some plastic fibers. So imaged firstly in low vacuum mode. We're just collecting a backscatter electron image there. And the other ones are gold coating with secondary and gold coating with a backscatter electron image. Now you will notice that we have some Z contrast here in the environmental thing, but we lose it, quite a lot of it, because we've done gold coating. So we've put on a very heavy metal. It's not very thick, it's nanometers, but it has obscured some of the detail we have when we're looking at average atomic number. So we'll have the carbon-based, carbon-oxygen-based polymer, and we have some particles of, in this case, some potassium, probably hydroxide or carbonate on the surface here. Here we can see an example of nanostructures. So here we're doing some tilting. So we can do some dynamic focus control to sort of extend the range of focus. We have things tilted so that we can see, if we looked at this top down, you wouldn't get any sort of idea of what's happening with the structure. So tilting is quite useful. Also, tilting can help us with getting more secondary electron detection. If you're tilting towards the detector, you can get more secondaries for doing imaging on things. Here we can see a fractured sample from tensile testing that some of the students have done for one of their labs. And here we can see both the secondary and the backscatter. So in this case, we're working at a longer working distance. So the sample is further away from the pole piece, so we can get a lower magnification, but also so we can get more sort of depth of field in the material. So we want to be able to have in focus a lot more of the sample. Here we can see some biological material. So with the fruit fly here, you can see in the secondary electron image that we are getting some charging. So there's some banding that is showing up. It's not terribly severe. So we could do, it is gold coated. It's sitting on conductive tape. So probably the problem we're having here might be a case of the gold coating was just sort of top down, and we're not necessarily getting a connection to the conductive tape, which is on the stub, which is in a holder, which is on the stage. So we're having a problem with the conductive path. So we are getting a little bit of charge building up despite it being coated with gold. So you could do things like maybe staining it instead, or use a coating machine which can tilt the sample so we can maybe get some side coating, make a path. You could also possibly use some paste to sort of join up the top of the sample to the holder or your substrate. Or you can do some adjustments of the parameters or go to environmental or low vacuum. So in this case, we can image sort of the eye here. So what we've done here is we've actually gone from a larger probe current to a lower probe current to sort of reduce the charging, to sort of image the finer sort of structures in the material. Here we can see polished cross-section mounted in plastic, so both secondary and backscatter. So we're looking at sort of the structure of the material. So some of the preparation you might have to do might involve doing some alterations to the material. So you have to make sure that you're not introducing some artifacts that you're going to misidentify certain things within the material. So a few references here. Very good book is Goldstein's book. The Australians have a very good website, which I would really recommend to people looking at. It does cover more things than SEM, quite a lot of microscopy on it. It's very useful for the beginner to sort of go to. There are some good sort of programs you can use for Monte Carlo simulations and doing some stuff with EDS. Some references about some things concerning SEM. So please don't use Q&A. Please use chat for questioning. So we're ready for doing some questions. If Sam can read them out to me,
yep.
We'll answer them as we go.
We've had quite a few questions, so we'll answer 3 or 4, and then we'll follow up with the rest of the Q&As in an additional video in the next week or so. Sorry, Chris?
Yeah, so also, I don't mind answering questions. You want to email me. So if you look up on our CCM website, you can contact me. It's Chris Butcher. I'm butcher@mcmaster.ca. So I don't mind answering questions. So it doesn't matter whether you're from industry or academia. or just general sort of interest, I have no problem in answering questions.
Okay. Okay, so first question, to do electron lithography, do we need a dedicated SEM or any conventional SEM can be used?
So the big problem with doing EBL, you have to have a system that allows you to do the resolution that you want to do. So you have to have things like a field emission microscope, unless you're doing things that are in the micron sort of size. If you want to do things that are a nanometer resolution, you have to have a very high-end field emission microscope in order to get a beam size that's small. You also have to have software that lets you control the beam to actually do the patterning that you want to do on the material. So we do have a system on our one of our field emission microscopes. It's a fairly low-end system. The biggest problem we have with it is the stage we have is not piezoelectric. It's a mechanical stage. So we do have some sort of backlash issues for doing really high resolution stuff. So it sort of limits us to the field of view that we get in the SEM. So we can't really do very large sort of structures. So it really depends on what sort of stuff. Just a second here. It really depends on what sort of things you're going to be doing. There are dedicated machines which will give you the higher resolution, which have all the components necessarily for doing fairly large patterning.
Okay, next question. What is the difference between the Magellan, the Joule 6610, and the Joule 7000?
So in the case of the, we'll start from the bottom. The Joule 6610 is a machine which you can use in both high vacuum and low vacuum. It's also a tungsten machine. So the size of the filament will produce a fairly large electron beam. It also doesn't have all the optics that some of the higher end systems have. So you have limited resolution due to the gun and the lenses that you have. But you can use it in low vacuum mode. So it does have the advantage that you can look at things that may charge up very easily, like non-conductors, or things that might have a little bit of bonded water, or limited sort of stuff that's sort of biological. The 7000 is a field emission microscope, so with the hot filament, much higher resolution. Because you have a lot more brightness with this particular gun, you can do a lot of low voltage work. It does suffer from the problem that the vacuum system, because of its age, is not as great as some of the more modern microscopes. But you can go up to 100,000 times magnification with sort of no problems. You have to do a little more tweaking above that. In the case of the Magellan, it's a machine that's designed for going up to really high magnification. So if we go back up to our very first sort of image, we can see an image here taken with the Magellan. It has a lot of different sort of adjustments you can make on the system. In this case, we're working at 700 volts. We're at very low voltage, so we can get a lot of surface detail. We've also put a bias on the stage, so the landing energy of the electrons is very low. So we're getting extremely great surface detail. We're also using a through-the-lens detector for improved sort of resolution. So we're up about 800,000 times magnification. And we're working at a very short working distance. So all these things combined we can get close to 1 million times magnification doing things. Also, why you see a black band at the top is because we're also doing some software correction where we're doing drift correction. So we're actually keeping track of when we're doing the imaging. We can actually image things. We can also do things where we're collecting the image in very fast frames and combining them together to actually get the image that we want for the sample.
Okay, what is the best practice / material for coating biological samples at high vacuum condition, and any tips for imaging?
So one of the problems you get with coatings is how visible the coating is. So it really depends on what sort of magnification you need to go to. So if you're not going above 30,000 times magnification, gold, platinum, gold, palladium, chromium, iridium can all sort of be used. Gold is the most common. Carbon, not as good sometimes as the metals for coating. As I said before, though, it's important that you have a connection to ground for doing things. So it really depends on what sort of magnification you're going to. So we tend to use platinum, iridium, chromium at sort of higher magnification. So if you're going up to sort of less than 100,000, those would be the ones that would be employed. You can also do some other things like staining, sort of with osmium tetroxide, ruthenium tetroxide of the actual biological sample to make it conductive. You have to worry about staining sometimes depending on the type. So if you're using a tungstenate staining, you might actually get crystals at higher magnifications that are visible. So there's various things you can do. It's important, though, that you properly dehydrate the sample. So you can see that you have to get rid of the water and not change the structure of the material. So it's important to properly dehydrate is the biggest thing. Coating-wise, if you have iridium or gold, palladium or chromium, that's a good choice to do if you have to do things above sort of 30,000 tons.